Have you ever really stopped to think about what's going on in your brain when you fall asleep? It's not just this passive state of nothingness. Sleep is an incredibly active, highly organized, and absolutely essential biological process. It's critical for our survival and our ability to function. So let's dive right into the intricate neurocontrol that governs this fascinating state.
All right, here's our roadmap for this explainer. First, we're going to define what sleep actually is from both a behavioral and a biological perspective. Then we'll get into its crucial functions, the why we sleep. After that, we'll examine the two-process model that regulates our sleep cycle, and then look at the brain's incredible flip-flop switch that gets us in and out of sleep. We'll take a journey through the different sleep stages, and finally, we'll connect all this physiology to real-world sleep disorders and the drugs that affect them.
So, where do we even begin? How do scientists actually define sleep? Well, it's not just one thing. It's really characterized by two key aspects. First, there's a set of distinct, observable behaviors. And second, there are very specific patterns of electrical activity in the brain that we can measure with something called an electroencephalogram, or EEG for short. What's really fascinating is that these behavioral hallmarks are incredibly consistent across almost all species, from us to fruit flies. First, you've got quiescence, which just means stillness. This is usually coupled with a specific posture, like lying down with our eyes closed. Critically, there's an elevated sensory arousal threshold. This means it takes a much stronger stimulus, like a really loud noise, to get a response from us. It effectively disconnects us from our environment. But, and this is a big one, unlike other states of unconsciousness, sleep has rapid reversibility. You can be woken up pretty quickly. And finally, sleep is governed by a homeostatic rebound. That's just the technical term for that undeniable urge to sleep longer to make up for lost sleep.
This table really helps highlight what makes sleep so unique. Let's compare it to a few other states. Think about a coma or a general anesthesia. Sure, they involve reduced responsiveness, but you can't just snap out of them. They're missing that rapid reversibility. And your body doesn't try to make up for lost time under anesthesia. There's no homeostatic regulation. Hibernation is kind of similar. Only sleep has this very specific combination of all three properties: reduced responsiveness, the ability to wake up quickly, and a biological drive to catch up when we miss out.
Okay, now that we know what sleep is, let's explore why it's so important. When you think about it from an evolutionary perspective, entering a state of reduced awareness is incredibly dangerous. It leaves an animal completely vulnerable. So sleep must be performing functions that are so vital that they're simply incompatible with being awake. And most theories point to sleep's critical role in supporting the central nervous system.
We can basically group the functions of sleep into two main categories. First, there's maintenance and recovery. This includes things like energy conservation, regulating hormones for growth and repair, reinforcing our immune system, and, really importantly, clearing out metabolic waste from the brain. The second category is all about enhancing our brain's computational power. This involves fine-tuning the strength of our neural connections and consolidating memories. And make no mistake, this is not optional. Prolonged sleep deprivation is literally fatal. It leads to a whole cascade of problems: a disrupted metabolism, impaired thinking, a weakened immune system, and, eventually, death.
So, let's start with energy conservation. How much energy do we actually save by sleeping? Well, during sleep, our overall metabolic rate decreases by about 5 to 10%. At the same time, our core body temperature drops by about 1 degree Celsius. Now, that might not sound like a huge amount for just one night, but over a lifetime, this adds up to some very significant energy savings.
Now for one of sleep's most incredible functions: waste removal. You can literally think of it like a dishwasher cycle for your brain, and it's managed by something called the glymphatic system. Here's how it works. During sleep, the space between our brain cells, that's the interstitial space, it actually gets bigger. This allows cerebrospinal fluid, or CSF, to flow more freely through the brain tissue and wash away all the toxic metabolic byproducts that build up during the day. As you can see here, the clearance of amyloid beta, a protein that's strongly linked to Alzheimer's disease, is significantly increased during sleep. This really suggests that poor sleep as we get older could make the brain more vulnerable to this devastating disease. This brings us to another really powerful idea: the synaptic homeostasis hypothesis. Think of your brain like a gardener, tending to this vast, complex garden. While we are awake, our brain is constantly learning, and this strengthens the connections, or synapses, between our neurons. But this growth is super energy intensive and just can't go on forever. So sleep acts like a master gardener. It intelligently downscales or weakens a lot of these connections. This conserves energy and, crucially, makes room for new learning the next day. It's like it protects the most important circuits while pruning back the weaker, less essential ones.
So, we've covered what sleep is and why we do it. But how does our body know when to sleep? Well, the timing and duration of our sleep are governed by this beautiful interplay between two core biological processes. It's often called the two-process model.
These two processes are called process S and process C. Think of process S, the homeostatic process, as a sand timer. The moment you wake up, that timer gets flipped and sand starts to accumulate. The longer you're awake, the more sand fills the bottom, and that increases your desire to sleep. This is your sleep pressure. Now, working right alongside it is process C, the circadian process. This is your internal master clock. It's a built-in, nearly 24-hour rhythm that promotes wakefulness or sleepiness at regular times, pretty much regardless of how much sand is in your time.
Let's look a little closer at process S. What exactly is causing this sleep pressure? The main driver is a molecule called adenosine. Our brain cells use a molecule called ATP for energy, and as that ATP gets used up, adenosine is produced as a byproduct. This adenosine builds up in the brain and it acts like a dimmer switch. It binds to receptors on our wake-promoting neurons and basically stops them from releasing their neurotransmitters like acetylcholine, noradrenaline, and serotonin. This widespread inhibition is what we physically feel as that mounting sleep pressure. And guess what? This is exactly how caffeine works. It's shaped just like adenosine, so it fits into the same receptors and blocks them, preventing the real adenosine from turning down that dimmer switch and making you feel tired. Now for process C, our internal clock. This master clock is located in a tiny region of the hypothalamus called the suprachiasmatic nucleus, or SCN. But how does it stay in sync with the outside world's 24-hour day? The key is light. We have these special cells in our retina called melanopsin-expressing retinal ganglion cells. They basically act like light meters. They detect the ambient light levels and send that information as a signal directly to the SCN through a dedicated pathway called the retinohypothalamic tract. This light signal is what entrains or synchronizes our internal clock to the external day-night cycle.
So what's actually happening inside these SCN neurons to generate this rhythm? It's this beautiful molecular dance that works like a negative feedback loop. Here's the breakdown. Two proteins, perfectly named clock and BAML1, pair up and act as an on switch. They activate the transcription of two other genes, period or per, and cryptochrome or cry. As the per and cry proteins are made and build up in the cell, they pair up and act as an off switch. They travel back into the cell's nucleus and inhibit the activity of that clock/BAML1 pair, which shuts down their own production. Then, over time, the per and cry proteins degrade, the inhibition is lifted, that on switch is flipped back on, and the whole cycle starts all over again. This entire loop takes, you guessed it, about 24 hours to complete.
Okay, so we've got the sleep pressure building up and the internal clock telling our brain when it's time to sleep. Now let's explore the actual neural circuits in the brainstem and hypothalamus that generate these distinct states of wakefulness and sleep. You can think of it as the brain's master switch.
This mechanism is often called a flip-flop switch, and this diagram shows you exactly why. We have two key groups of neurons that are in this constant push-pull relationship. On one side, on the left, you've got the wake-promoting centers, which we collectively call the ascending arousal system. On the other side, on the right, is the sleep-promoting center, the ventrolateral preoptic area, or VLPO. And here's the key: they mutually inhibit each other. When the arousal system is active and winning, it's releasing neurotransmitters that shut down the VLPO, and we're awake. But when the VLPO becomes active, it releases an inhibitory neurotransmitter that shuts down the entire arousal system, and we fall asleep. This mutual inhibition is why the transitions are so rapid and the states are so stable. You're either firmly on one side or the other.
So let's meet the key players in this battle. The arousal system is a team of brain nuclei that use a whole cocktail of neurotransmitters to keep us awake. Norepinephrine from the locus coeruleus, serotonin from the Raphi nuclei, dopamine from the PAG, and histamine from the TMN. The sleep-promoting system in the VLPO uses the brain's main inhibitory neurotransmitter GABA to silence all these arousal centers. But there's a crucial third player, orexin, which is also known as hypocretin. Think of orexin as the coach of the arousal team. It's produced by neurons in the hypothalamus that provide strong excitatory input to the arousal system, reinforcing that wake state and preventing the switch from flipping at the wrong time. All right, so the flip-flop switch has flipped us into sleep. What actually happens next? Well, sleep isn't just one uniform state. Instead, we cycle through different stages, and each one has its own unique brain activity and purpose. This whole progression is known as sleep architecture. So let's take a journey through a typical night.
This chart here is a hypnogram, and it maps out our journey. You can see time along the bottom and the sleep stages on the side. We start in wakefulness at the very top, and then we descend into non-REM or NREM sleep. Stage 1 is that light, drowsy period. In stage 2, we drift a little deeper. Then we hit stages 3 and 4, which are the deepest stages of sleep, often called slow-wave sleep. You can see on the chart, this is when the brain waves are at their largest and slowest. After we hit this deepest point, we actually ascend back up through the lighter stages, and then we enter a completely different state, REM, or rapid eye movement sleep, which is marked here in red. This entire cycle takes about 90 minutes, and we repeat it multiple times throughout the night.
So, what's happening at a cellular level to generate those slow waves of NREM sleep? The key lies in our thalamocortical neurons. You can think of the thalamus as a gatekeeper for sensory information on its way to the cortex. When we're awake, these neurons are in a tonically active state. The gate is wide open, and sensory information flows freely. But during sleep, they switch to an oscillatory or bursting mode. The reason for this switch is a change in the properties of special ion channels, called low-threshold T-type calcium channels. This switch creates a rhythmic, synchronized oscillation that effectively closes the gate, disconnecting the cortex from the outside world and allowing the brain to focus on its internal jobs.
One of the most bizarre and important features of REM sleep is atonia. That's the near-complete paralysis of our voluntary muscles. This is a brilliant and totally essential safety mechanism that prevents us from acting out our often vivid dreams. And here's the brain's precise 3-step plan to achieve this, all starting at a part of the brainstem called the pons. Step 1. A group of GABAergic neurons becomes active and inhibits the wake-promoting noradrenergic and serotonergic neurons. Step 2. This is cool. It releases the brakes on a population of pontine cholinergic neurons, which then activate a group of glutamatergic neurons. And finally, step 3, these glutamatergic neurons project down to the spinal cord, where they excite inhibitory interneurons that use the neurotransmitter glycine. These interneurons then directly inhibit our motor neurons, preventing them from firing and causing that paralysis.
This table does a fantastic job of summarizing and comparing the 3 major states of consciousness. Let's look at the contrasts. During NREM, sensation is dull or totally absent, but during REM, it's vivid and internally generated. That's our dreams. The most striking difference, of course, is movement, with that muscle paralysis being totally unique to REM. But look at the EEG. The brain activity in REM sleep, low voltage and fast, it looks almost identical to the waking state. This is exactly why REM sleep is often called paradoxical sleep. The mind is incredibly active, but the body is completely still. It's a truly unique state.
All right, for our final section, let's connect everything we've learned about the intricate machinery of normal sleep to see what happens when that machinery breaks down, like in sleep disorders, and how drugs can be used to manipulate these very same circuits.
Let's make the connections. In narcolepsy, patients experience this overwhelming daytime sleepiness and can suddenly just collapse into sleep. We now know this is caused by the autoimmune destruction of those orexin-producing neurons. Remember, the stabilizers of the flip-flop switch. Without their coach, orexin, the switch becomes super unstable and can flip at totally inappropriate times. Stimulants like caffeine and Ritalin promote wakefulness by targeting the systems we've talked about. Caffeine blocks adenosine receptors to fight that sleep pressure, while Ritalin stops the reuptake of arousal neurotransmitters like dopamine and noradrenaline. And on the flip side, sedatives work on the other side of the switch. Benadryl blocks the arousal effects of histamine, while drugs like Valium enhance the inhibitory effects of GABA. Finally, in the rare and tragic disease, fatal familial insomnia, a prion disease causes massive neurodegeneration in the thalamus, destroying the very neurons that generate our sleep rhythms. This deep understanding of sleep circuitry is now paving the way for a whole new generation of highly targeted drugs, like orexin receptor antagonists for insomnia. But it just makes you wonder, what other secrets of sleep are waiting to be revealed? The journey into the sleeping brain really has only just begun.